Crystal, if you could pull up the first slide, we're going to get going. Okay, so it's 12 o'clock and we're going to start this workshop. This is the Synthetic Cell Workshop Reflections on NSF's Understanding the Rules of Life program. Next slide, please. So as I mentioned, this is the Synthetic Cell Workshop. It's happening today. This is third of 4 workshops that the National Academies organized at the request of the National Science Foundation. So we're having the last one next week. So today it's the synthetic cell. So we are gonna have PIs of synthetic cell projects talk to us. My name is Camila Abels, by the way. I am the responsible staff officer for this workshop series. So registration and other information about these events are posted at the project website. And if you have any questions about upcoming events, please contact Christine. Her email address is here on the screen. And I also just want to say, please be advised that this workshop will be recorded. And materials presented, slides, verbal comments, and comments submitted via Slido may be used in the workshop proceedings with attribution. So now I am going to turn it over to Corey Wilson, who is one of our moderators.
Next slide. I'm Corey Wilson. I'm a professor at Georgia Tech. Prior to my faculty appointment at Georgia Tech, I was faculty at Yale. In fact, I shared at least some affiliation with Farron. I think he was located at the West Campus when I was there. So we've been charged to run this workshop and get some feedback on this synthetic cell slide. I was funded with the inaugural set of Understanding the Rules of Life proposals prior to the 3 sets of iterations that you see currently. So I'm really excited about this. And so we can go to the next slide. I'm joined by Kate and Eric. They're my moderators or co-moderators. I think that they are going to bring interesting perspective in terms of bottom up to top down discussion with regard to synthetic cells. So I'm excited to have this discussion with them. To the right, you see our discussants. So Alan, Chris, Jeff, who will join us around 1:30, I believe. Sydney, Mary, and Farron are actually going to be sort of the guest of honor who are going to facilitate much of the discussion that we're going to have today. Next slide.
Oh, Corey, can I just make a correction? Jeff is here and he's leaving at 1:30.
He's leaving at 1:30. Okay.
No, actually, I'm good. I'm good the whole time.
Oh, great. Wonderful. Okay. Thank you.
Even more corrections. Good. So, all right. So this is the agenda today. Again, we're going to have brief introductions, workshop goals, etc. And then we're going to transition into the bulk of the discussion, which will be the interactive discussion with regard to rules of life with the principal investigators. Take a break, and then we'll reconvene around 2:30, and then we will wrap things up. I'll turn it over to Eric.
All right, thanks, Corey. So just to give a quick overview of some of the goals for the workshop, in about 4 slides from now, we'll talk about more specific expectations for the workshop. But in terms of just general goals, what we're going to talk about today are some of the scientific advancements the PIs have made during the funding periods, as well as talk about how interdisciplinary research has allowed you to attain some of your research goals and maybe how it hindered your approach towards your research goals. We'll also talk about some future societal needs and scientific questions that you all tried to address during your funding periods. And then if we're lucky, we can highlight some rules of life, maybe beyond the obvious, which is to survive, that may be generalizable across fields and across various scales. So next slide, please. So just for perspective, in 2016, the National Science Foundation came up with 10 big ideas that were implemented in order to position our nation at the cutting edge of global science and engineering by bringing diverse disciplines together. One of those big ideas was understanding the rules of life, which you all know is based on developing predictive understanding of how key properties of living systems emerge from interactions from factors such as genomes, phenotypes, and the varying environment that all life lives in. So funded projects have addressed rules at multiple levels by addressing fundamental questions in life science that cross different scales and produce broadly generalizable results to permit formulating a quote unquote rule and ultimately enable prediction of change in all biological systems. So next slide, please. So some of the key programmatic goals for understanding the rules of life. One is to enable discoveries that will allow us to better understand interactions or how we generalize these rules. Another one is to provide us with the capacity to approach more complex questions, meaning how do we build a broader and more robust foundation upon which to ask more sophisticated questions. Also, we want to train the next generation of researchers to approach scientific inquiry in a way that, as I mentioned, crosses scales and disciplines. And lastly, we want to foster collaboration and convergent research to consider multiple levels of organization and complexity in addressing key questions in the life sciences, as well as, of course, engineering. So next slide, please. So, and although a outcome towards understanding these rules is to build various synthetic cells focused on transforming our understanding of the molecular mechanism that underscore the building and function of systems that reproduce traits and qualities about living systems themselves. Okay, so with that next slide, I think Kate's going to talk about some of the workshop expectations.
Yes, thanks, Eric. So we'll have a discussion on the 3 key topics that were driving this program. So we'll talk about the scientific advances that this research enabled, and we'll discuss the multidisciplinary aspects of those projects, including all the issues in education and training that the PIs might have run into. And then we'll talk about the societal issues that were addressed during the research in all the teams. And we also want to identify ways in which we can help the NSF to improve the next rounds of this program. And if you're watching this as a webcast, please use Slido to ask your questions. And I think Jess will run us through the logistics of that now.
Thank you, Kate. Yes. So Slido is our platform that we are using today for receiving your comments and your questions. If you are on the registration page through that you received via email. So it should be right below the actual browser that you are using to watch this. And so if you put in your name and click that you accept the terms, and as you join the event, then you should be able to see where you can send in your questions. We would love if you could put in your name and affiliation just so that we know who to attribute it to, as we will be asking questions throughout the workshop. And also just saying this is similar to the workshop in general. As we are recording, we are also using all of these public comments. Anything that you send via public comment could potentially be included into any of our deliverables that we end up publicizing. We'll send them online. If you are somebody who is joining this meeting through the Zoom link, so that'll be our discussants and our moderators. If you have questions, feel free to put them into the Zoom chat and we will be able to ask them there. And next slide, please. I'm passing it back to, I believe, Kate.
It's going to
Corey.
Kate. It's going to
Yeah,
Kate.
thank you. Okay, so the PIs were given an opportunity to answer some questions about the scientific discoveries and challenges that they encountered during this program. So the next few slides will be kind of a wall of text that I will try to summarize for you, but I might just stay quiet for about 10 seconds just to give people a chance to read through those. So let's move on to the next slide. So now we're going through basically answers to those scientific results or challenges questionnaires that the PIs in this program were asked to fill. So one team was working on engineering minimal neurons, and they were identifying genes that were needed to build the simplest cell that has some but not all functions of neurons. And that included identifying a minimum number of channels that are needed for neuronal activity. And then another team expanded our understanding of what is needed to create ultra-fast contracting cells. And just take a couple seconds to read the replies. All right, let's hope people read fast. Let's move on to the next slide, please. Thank you. So another team worked on engineering a ribosome capable of translating amino acids of opposite stereochemistry. And they were learning how to better recode the genetic code in general as a result of that work. Then another team was working on the relationships between the physiology of the synthetic cells and the size of the cells. So they were basically investigating those scaling relationships in metabolism. And then another team was investigating wound closing, and they discovered a novel mechanism of cellular signaling, generally helping us better understand the mechanical properties of living cells. And again, a couple seconds to let people read through the slide. All right, let's move on to the next one, please. So another team was investigating RNA genomes of viruses, and they made some interesting discoveries in structured sequence relationship, very relevant to our current situation. Another team worked with CRISPR, because it seems like these days it's obligatory that there is a team that uses CRISPR in every project. And that team discovered a new regulatory mechanism for gene expression that can be used for engineering synthetic cell gene regulatory circuits. Another team worked on coacervates, and they were investigating the mechanisms for formation of membraneless organelles. And you can read through those slides now. Okay, so now we'll move on to more of a summary. Next, please. So our discussion, these will be the kind of main topics that we'll cover in our discussion. So the kind of a main advancements out of this program were in engineering synthetic neurons, the mechanical forces that are moving cells, the principles guiding structure-based recognition of RNA in viral genomes. And next one, please. Other topics where our understanding of the limitations of the chirality of translation and how to reprogram the ribosome to accept the orthogonal chirality monomers, and then mechanistic understanding of cells and the scale relationships in biochemistry. And if anyone has any questions, you can speak up, or I can hand it off to Eric to talk about the multidisciplinary aspects of this work.
Yeah, Kate, before we get into PI responses to the scientific advances in rules of life, Corey, I think we forgot to have the PIs introduce themselves.
Sure, we can do it.
So if we could do that before we begin the discussion, that would be great. I guess there are 6 PIs.
Yeah,
we
I can moderate that.
go down the list and have each, from my perspective, it'd be nice to know.
Yeah.
So
So,
each PI introduced the team that they had, like what 2 sentences, what was the goal of the project?
Yeah.
And what PIs were collaborative in terms of science and engineering.
Sounds good. So we can start with Alan. If you can go ahead and do a brief introduction. Try to keep it about at a minute and a half, 2 minutes.
Sure. Hi, everybody. My name is Alan Liu, faculty in mechanical engineering and biomedical engineering and biophysics at University of Michigan. So I lead a team on building synthetic neurons and synthetic neuronal network. You'll maybe hear a little bit more about that later. What we're trying to do is to come up with the minimum set of ion channels that may be able to reproduce an actual potential. But we're also interested in engineering living neurons to have different forms of communication between, say, two non-neuronal cells and behave like neurons. So that's sort of the gist of our project. And this is a team of 7 PIs. So myself at Michigan, and we have Cindy Tang from Stanford, who is also a discussant, TJ Ha from Johns Hopkins, Chong-Ling Wang from Purdue, Mo Das from Rochester Institute of Technology, Barbara Harthorne, a social scientist from UC Santa Barbara, and Francois Saint-Pierre at Baylor College of Medicine.
Yeah, outstanding. Thanks, Alan. Chris, can you go next?
Yeah, thank you. I'm Chris Kempis. I'm a professor at the Santa Fe Institute. I'm a theoretical physical biologist. And so I think a lot about simple theories that allow us to take the vast diversity of life and project those onto simple axes or extract general laws looking across the diversity of life that we have on Earth. I worked on a team with Kate Adamala and John Glass, thinking about constructing a synthetic cell from non-living parts. And in that role, I ran the theoretical portion. So basically trying to think about what are the design constraints when we think about something like a bacterial cell that really allow us to do engineering. And so that involves a lot of physical constraints, as well as some emergent evolutionary and developmental constraints for cells, to think about what is really the main trade-off space for a synthetic cell or for the physiology of small organisms. Thank you.
Okay, thanks, Chris. Mary, can you go next?
Yeah, hi, I'm Mary Elting in a physics department in NC State. And the goal of our team was to build synthetic cells, or is to build synthetic cells at the mesoscale. So sort of neither top down nor bottom up. But by starting with living cells and making them more synthetic by adding synthetic parts, beginning with synthetic cytoskeletons. And our team includes a physicist, a chemical engineer, a chemist, cell biologist, as well as a historian of science. And those are myself, Saad Bamla at Georgia Tech, Fred Chang at UCSF, Aaron Dinner at the University of Chicago, and Jane Meinstein at Arizona State and the Marine Biological Laboratory. Those were sort of the initial members of our team, and then we added 2 additional PIs, Scott Coyle at University of Wisconsin and Jerry Hans at Drake University.
Okay, thank you, Mary. Cindy?
Hi, everyone. I'm Cindy Tang from Stanford. I'm in the Department of Mechanical Engineering. Our project is to study the wound healing mechanisms in the giant single cell free called Stanters aureus. This project is NSF. The collaborator is Professor Wallace Marshall at UCSF. It's in biochemistry and cell biology department.
Cindy, your connection was bad, at least on my end, but just FYI.
Oh, okay. All right. I'll try to see if I can fix it. Sorry, it's raining in California, PG&E.
No
anyway,
problem. All right, Farron, can you wrap us up?
Sure. Thanks, everyone. Farron Isaacs from Yale University, where I'm part of the departments of molecular, cellular development biology, and biomedical engineering. I was been part of a team that came from an ideas lab, where a number of others on this call have come from, working on a project entitled Booting Up a Mirror Cell. Involved in this project is Neil Devaraj, a co-PI, Kate Adamala, who's with us today, Irene Chen, Jonathan Carr, and John Evans. who's been involved in some of the LC components of our project. In brief, we've set out to effectively see if we can engineer and produce biomolecules that have their mirror chirality. So, for example, in nature, you've got denucleic acids and L proteins. One of our goals is to be able to chemically synthesize D proteins and encode D amino acids at the ribosome. So looking forward to participating in today's workshop.
All right, thank you, Farron. Okay, I'll kick it back to Eric. We'll get us started.
If I may,
Jeff, you go next.
Oh, sorry about that. You're not on my existing list, Jeff. Sorry.
all right. Yes, I was late. I was late in responding. I think in some way I'm involved in this program in 3 different ways. Excuse me. I've been running the international SC 2.0 project with the synthetic yeast genome, which has been funded by NSF and pointedly not by NIH since its very inception, and it long predates the Rules of Life program. But I'd like to think it was an inspiration for it. It involved a very diverse group of collaborators all over the world. But the major co-leader of it was Joel Bader in biomedical engineering at Johns Hopkins. I myself am a geneticist. That's what I consider myself here at NYU. And we started this project more than 15 years ago with a small seedling grant from NSF, and it grew over the years. But so there's lots of interesting things to say about that. We definitely had to take an interdisciplinary approach, a computational approach to genome redesign, so to speak, as well as work out a lot of the technology to make it feasible. In terms of the UROL program, we participate in 2 grants. One was from the Ideas Lab, and that's the RNA genomes and trying to see how big of an RNA genome can we make. And that too was done primarily with Dr. Bader at Hopkins, but also includes prokaryotic geneticist Kai-Han Wang from Caltech and Deborah sorry, blanking on her last name right now, bioethics at Johns Hopkins, and a filmmaker. And so we've had a lot of fun working on the genome of this very small and simplest virus called a narnovirus, and trying to see if we could grow it into something much larger, like basically an RNA chromosome. And happy to talk more about the successes and failures in that. And then the second thing is we have a larger UROL grant on humanizing the chromosomes of yeast, where we've been able to completely replace substantial components of the chromosome, namely the histones, with the human counterparts. And we've started projects to humanize other aspects of chromosomes, key components like the kinetochore, which with substantially less success. But anyway, happy to talk about all of that. And that the major collaborator there is Matt Morano, a computational genomicist here in our institute. Thank you.
All right, thank you, Jeff. All right, now we're going to go back to Eric. Okay.
Yeah, Corey, I don't think it's my turn yet. Right? If I understand the format correctly, aren't we supposed to have a discussion with all the PIs on Kate's last 2 slides?
That's kind of what I thought that we'll just basically ask people now to go through the slides and have a discussion before we move on to the multidisciplinary part.
Yeah, okay. So Kate, do you want to direct questions towards specific PIs, or do you just want to pick a PI and have them tell us what the scientific accomplishments were during the funding period?
Can we have the slides back, please? Maybe they'll help. And yes, I'm happy to point, but if someone wants to go first, maybe that's easier to have a volunteer. If someone would like to go first and basically talked about what his or her team found, especially something unexpected that you found during this project.
Yeah, so we just want to spend like the next 20 minutes or so, 20, 30 minutes talking about the major scientific achievements for each of the 6 groups present here today.
Well, I'll be happy to start, since I see you've got the RNA viral genome up here. So as I said, the project that we're doing with Dr. Bader, Dr. Wang, and others revolves around an idea that was pitched by Dr. Wang and Dr. Bader at the Ideas Lab, which was essentially how big of an RNA chromosome could you build from scratch? And so Dr. Wang decided to pursue building RNA replicons in bacteria based on bacteriophages and based on some really old work by Saul Spielman on replicons that emerge in vitro transcription reactions. And we decided as our starting point in my group to work with these related replicons found in yeast strains and certain other species called narnavirus, which stands for naked RNA virus. And these are quite small, about 2.5 kilobases. And our goal was to try to get to replicons of 50 or more kilobases. And I have to say that I was totally wrong in my choice of elements, because we have not succeeded in lengthening the genome of that element pretty much even by more than 10 bases or so. And what we discovered, which we completely didn't expect, was that unlike other viruses, which tend to have special secondary structures that define the termini of the virus or start replicating here, this class of viruses is very highly structured throughout. So we call it pervasive RNA folding. And what we've shown, again, to our surprise, is that this pervasive RNA folding is crucial just to maintain the RNA from being degraded in the cell. So if we recode even relatively short segments of the open reading frame that's encoded by this virus that's required for its replication, not only is the virus completely unable to multiply, we can basically not detect its RNA in the cells where we're driving its transcription with a very strong promoter. So, of course, we still want to try to hit our goal of making a much larger replicon than this. And of course, we noticed during the pandemic that there was a virus that we all have gotten to know very well, which is made of RNA and which has a genome of 30,000 plus bases. that perhaps it would be possible to make larger replicons. And so we are now working with a mammalian RNA replicon based on the Sendai virus, and we are attempting to grow that to much larger than its native size. And that's a quick capsule summary of our progress. And let me just say that absolutely crucial to coming to all of these conclusions was our collaboration with Joel Bader, who did tremendous job of analyzing and coming up with new ways of visualizing RNA secondary structure predictions for what has now become quite a diverse clade of viruses that has multiple interesting subgroups with different biologies.
Can we drop the slide so that we can see the discussion? Yeah. Okay.
Hey, Jeff, I had a question about your project. So we know of RNA as an informational molecule as well as kind of a catalytic molecule.
Mhm.
And you're interested in it as an information molecule, but we also know a lot of catalytic RNA, large catalytic RNA requires binding of protein.
Mhm.
Do you think that could be necessary also for a large informational RNA molecule?
Yeah, it's a great question, and one of the sad truths of this very poorly studied family of viruses is that not very many studies have been done on it. There's basically been one lab in Spain that's been working on it for the last 20 or 30 years, and they developed this beautiful launching system that can launch the virus. We fortunately were able to debrief both of the PIs. As a husband-wife team, and acquire a lot of knowledge through a visit there with them. We learned from them that, it's in the literature that there's a one-to-one complex of the RNA and replicase, but no other host protein is known to be required for the replication of the virus. Of course, the translation machinery is absolutely essential, but that's presumably a generic requirement. So it's something we'd love to look into, whether there are other host proteins present in this complex, and that's one of the other projects we're pursuing now.
Okay, great. Thanks, Jeff. Kate, are you going to introduce Vincent first before Alan goes?
Yeah, I'm happy to ask Vincent to introduce himself.
Hello,
Sorry,
everybody.
we skipped you.
Yeah, no, because I registered very late, so I apologize. So my name is Vincent Noiro. I am a professor at the University of Minnesota in the physics department. And I am part of a team that is called the proteocell, the fat-free cell. And the idea here is still to build from the ground up a synthetic cell using the biomolecules. And without lipids, without fat, and especially without phospholipids. And this project is mostly based on liquid phase separation. That's it. And in the project, there is Cheryl Kerfeld at Berkeley, Barbara Arthron at UCSB, Giovanna Ghirlanda at Arizona State, Christine Keating at Penn State, and Professor Sullivan, Millicent Sullivan at the University of Delaware, and myself. That's it. We are 6 PIs. And we have made some relatively nice achievement. I don't know if I should talk now about it or later, but we were able to make liquid phase separation in a buffer, relatively simple system, and put proteins, and they they go at the interface of those droplets. And those proteins, they come from self-assembled structure that you find in nature, like BMCs, bacterial microcompartments. And another part of the project was to use a cell-free translation system so that we can do gene expression. And we were able also to do liquid phase separation and to start to colocalize also proteins at the interface to replace the phospholipid membrane. That's it. Thank you.
Thanks so much, Vincent. So any more questions before we move on to Alan?
All right, I guess I'll start then. Yeah, so I'm going to talk a little bit about the project, building a synthetic neuron and synthetic neuron network. Like all the PIs here, we were part of this workshop that took place in 2019 that at the time we just came up with really crazy ideas, and I'll say our ideas was definitely not easy. I'll describe sort of the ambition behind building a synthetic neuron. At the time, I was thinking, what cells could it build? So we are, a human is made out of 200 cell types, and in principle, we can go after any one of the 200 cell types. And I thought at the time, neuron has, electrical properties, basic ion moving across the lipid bilayer membrane, and would that be something that we can recapitulate as a functional aspect of a very important cell type in our body? So let me just sort of briefly describe what we're trying to, at least one aspect of what we're trying to do is to generate an actual potential, what happens in the axon. If you think about a neuron or a pair of neurons that, there's the postsynaptic neuron that basically is where the signal begins. And there is a ligand-gated ion channel that triggers the initial, voltage increase. So the depolarization. At the resting level is about minus 70 millivolt, and the initial kick basically puts it up to say minus 50-ish millivolt. That triggers the voltage-gated ion channel. So specifically the sodium voltage-gated ion channel that allows the sodium to enter the neuron, and this further depolarize the transmitting potential to kick in the voltage-gated potassium channel. So then now the potassium will leave the cell. That will then causes the transmitter voltage to go down. Finally, you trigger the voltage-gated ion channel at the nerve terminal that triggers the neurotransmitter release. So that's the sort of the basic picture that you can find in the textbook. And I've mentioned just about only 4 membrane transmembrane ion channels, right? So there's the ligand-gated channel, the sodium voltage-gated channel, the potassium-gated voltage-gated channel, and the voltage-gated calcium channel. So the naive thinking is that if we can reconstitute this minimum set of ion channels, we may be able to recapitulate actual potential. So that's the hypothesis going into this project. And because we're trying to build this from the bottom up, I'll talk a little bit about one of the advances that we were able to do is we need to be able to make these channels in the lipid bilayer membrane. So we utilize something called self-expression, which Vincent just talked about. Essentially, these are experimental techniques to produce a protein without using a cell, right? So you utilize a cell lysate, and specifically we utilize a HeLa-based cell-free system. So mammalian cell-free system. So if we supply a piece of DNA, we can basically make the protein of interest. Because we're making transmembrane protein is challenging, and I think as part of this rules of life, we may have I wouldn't say discover, is that in fact you can actually make a transmembrane protein without having a whole bunch of very complicated machinery that you find in the cell. So in our reaction, if we supply membrane, lipid bilayer membrane, and we use the self-expression system, we can actually we have evidence showing that these membrane protein will directly insert into the cell, into the membrane there. And this could be mediated through these ER-derived microsomes that are found in the lysate. Of course, one can argue the efficiency might be low, but we certainly do find evidence for this incorporations of membrane protein. And because we can do that, then we've shown that we can introduce multiple transmembrane proteins. So the channels I mentioned can become incorporated into the lipid bilayer. So one of the things that we're trying to do now is to bypass, or I guess to utilize sort of non-native channels or non-native ion channels for stimulating this action potential. So we're using a light-gated channel. So people might have heard of optogenetics, channel rhodopsin. So idea is to introduce these channels so we can stimulate this very artificial synthetic neuron by light and then triggering the action potential. So that's the aspiration. We're still very far from that. I think some of the challenges, if we get to discuss these challenges later, is actually very difficult to test some of these functionalities when you start incorporating multiple channels into the system. I think each channels need to be independently tested before putting them into a system. That's going to remain as a challenge for a project like this. Maybe I'll sort of stop there. So this is only talking about achievement from my lab and the bottom up. There's also folks on the team that's working on sort of introducing ion channel into native cells, and happy to talk more about that if there's interest.
So Alan, for the channels that you can incorporate into a lipid membrane, are they active, like in a biologically relevant manner?
Yeah, that's a great question, Eric. So that's where we're trying to spend a lot of effort testing these ion channels. So outside of this particular project, my lab has also worked with the mechanosensitive ion channels, and that channel is active because we can exert membrane tension by changing osmotic pressure, and we do show that channel is active. For these ion channels, we haven't had success in showing that they're active as of yet. So yeah, but that's going to be one of the next hurdle to really make sure those channels that we introduce are functional. We've shown this with other channels, but not these ion channels yet.
Yeah, I'm going to follow up on Eric's question. So Alan, what is the purpose of using a cell-free system? I mean, what are you gaining by using cell-free?
Yeah, definitely. Yeah, it's a great question, Corey. So, so traditionally, people work with membrane proteins with purify a protein from either mammalian cells, and that utilizes detergents. And when you reconstitute that membrane protein into the lipid bilayer, you inevitably would introduce random orientation of the membrane protein. It might not matter so much for certain membrane protein, but for some it matters, especially when you want to look at transmembrane potential. You do want to have the membrane protein all sitting in the same orientation. So the way we introduce our cell-free system is we actually encapsulate our cell-free reaction within the lipid bilayer vesicle. Mm-hmm. And the way that these proteins will be made would be ideally orientational specific, because it relies on sort of the same, well, hopefully the same way that transmembrane proteins are introduced. There are some papers that talk about like introducing signal peptide sequence, in front of certain genes, and that has shown certain specificity in the orientation. And we're using similar techniques as those that have been shown recently.
So, I'm sure you're aware of this. Cell-free systems are notorious for issues with reproducibility. So, is this more in the sense of, qualitatively, I believe you can get what you want, but quantitatively, does it matter that you're using a cell-free system? And if so, what are some of the things that you perhaps want to take into consideration when you're expressing these transmembrane proteins?
Yeah, that's another great question. So my lab started out spending the first 2 years developing a homemade mammalian cell-free system. We used that for about 5 years. And after that, student graduated and we lost that ability to make the mammalian cell-free systems. Now we actually go with fully commercial cell-free kits. And obviously then you're at the mercy of, the quality control of the things that you buy. We also use a pure system. So these protein, these recombinant, it's protein synthesis using recombinant elements. So the pure system is another system we've been using recently. And those are pretty heavily used across the field, and it is worth noting that there are reproducibility issue with these kits too. So we've seen batches that work and batches that don't work as well. Yeah.
Can I ask a quick question? I don't know if I can, but
yes, please do that.
Yeah, I can. So Alan, thank you. I just wanted to ask you a technical question. Have you tried all the kits possible that are commercially available? Some of them are higher organisms, they may be more suitable for membrane.
I've tried only the two. I mean, I know you have a kit that that we have them in our freezer. But there's also a plant based kit. We have some samples. We never done them side by side.
Nice.
But it may be worthwhile for someone to take all the available kits and just do a very clean study on that. Yeah.
Okay.
Yeah. All right. Thank you.
Thank you very much. If there are no more questions, Alan, I just want to say I love this project and I sympathize with all of those problems. If there are no more questions, we can move on to the next project, Mary.
Sorry, I was having trouble unmuting. I think it's working now. Excuse me. So like Alan said, our project also came together at the Ideas Lab workshop. And I think for our team, we really are working in a direction that was new to all of us, and also we were new to each other. So I think none of us on our team had worked together before, and most of us, I think, did not even know each other before the Ideas Lab workshop. So we're really kind of we've been starting from scratch in terms of building out our collaboration as well. And so the long-term goal of our project was this idea that I mentioned already, where could we build a synthetic cell, not by starting from the top down and stripping things away, and not by the bottom up and building things in one piece at a time either, but from sort of starting at the middle, introducing synthetic parts, and then adding more and more synthetic parts till you have something that is wholly synthetic in the long term. That was kind of the idea. And one of the questions that arises with this idea is, how do you get the parts within the cell, these synthetic parts, to talk to each other? And so as a starting point for addressing this question, we were thinking about force generation and cytoskeletal force generation. So how could we either put non-native, which we called, or non-biological parts into the cell, which we called prosthetic cytoskeletons, or parts that were biological, but that came from a different kind of cell, right? And then if we introduce those into a cell, how would they react to these biological but non-native parts, and how could we teach them to integrate with the rest of the cell? So that was kind of the question we started with, and we cast a broad net in terms of approaches to begin with. And so we explored this prosthetic idea mainly by building magnetic tweezers based devices and using magnetic particles inside cells to try to manipulate and generate force inside cells. And we made some headway on that, but the thing that took off more quickly was instead looking at force generation that is sort of non-canonical. So in terms of cytoskeletal force generation, networks that we know about, actomyosin contractility is kind of one of the main ones. There's also force generation by the microtubule cytoskeleton. And those are each fairly well understood and somewhat ubiquitous in most of the cells that we study. But in terms of the sort of evolutionary tree of life, there's a lot of different ways that cells generate force that we don't understand yet. Because our sort of approaches to biology have often focused on the parts of the evolutionary tree nearer to us, and including fungi or yeast who are actually our pretty close evolutionary neighbors as well. And so we began investigating the contractile machinery of some unicellular ciliates, which have this ability to contract to half their body length in under 5 milliseconds. It's one of nature's fastest known motors, and is very poorly understood in terms of the underlying biochemistry as well as the mechanics of how this process works. And so we've been focusing on better understanding this machinery so that we can eventually use it to add it into cells as a synthetic cytoskeleton. And so we've made a lot of headway on in vitro reconstitution of these myonin proteins from sporostomes, as well as building a theoretical model that can explain how does this contraction occur and what are some of the limiting factors in the contraction. So by doing ultra-fast imaging of whole contracting sporostomum cells, we can see from the mechanics of that process in a sort of normal buffer as well as in highly viscous solutions, that the sort of viscoelastic environment is one of the limiting factors, as well as the calcium wave that travels from one end of the cell to the other that triggers this contraction moving along the cell's body. And I should have said the other thing that's really unique about this machinery is that it's not ATP dependent. So unlike most of the other contraction that we know about, it's not powered by ATP.
Hey, Mary, can I ask a question?
Yeah.
When you say in vitro reconstitution, what do you mean?
Yeah, so the machinery that powers the contraction in these cells is not well described, and so we would like to have a purified protein or 2 or 3 that can, in response to some signal, generate force in purified protein form in the way that can be done for like actomyosin contractility, for example. But in this system, it's not based on actomyosin, and in fact, the components that are necessary and sufficient for that contraction are not fully known. So that's one of the things that we're trying to determine.
Is it a machine that moves itself or moves the cytoskeleton?
I would describe it as a cytoskeleton. I would say that it is a cytoskeleton. It's sort of a non-canonical cytoskeleton. So it's moving itself because that's one of the things the cytoskeleton does, right? It generates force, and then by being anchored to the rest of the cell, it moves the cell, with it. But that's another question: is how does it attach to the cell without pulling itself apart? I mean, the forces that these cells have to sustain are hugely greater than for example, muscle contraction, and how they are able to do that without ripping themselves apart is one of the questions.
Yeah. Okay.
Yes. So Mary, I'll follow up. So maybe a different way of asking Eric's question is, so what are your design goals?
Yeah, I mean, so I think it's sort of in the long term or in the short term. I mean, in the short term, our design goals, I would say, is to understand to generate a list of components that are sufficient for generating contraction in vitro, in a purified system. What's the minimum number of proteins that we can mix together in a test tube and add calcium and cause it to generate force on the in vitro side? I would say that's our short-term goal.
So once you have those parts, you can build what?
Then we can put them into a cell that doesn't normally have them and see if that cell can sustain the large force generation that those proteins can cause, right? So if I put those into a mammalian cell, which doesn't usually get contracted by a factor of 2 in 5 milliseconds, can I anchor them in a way that I can cause that mammalian cell to do this behavior that it normally can't do, or do I just rip it apart? This helps us understand sort of how can we imagine putting synthetic parts together in a cell that's more like our cells. Can we give our cells new abilities that they don't have to do by taking abilities from this other cell that's already developed them, or are there so many components to that system that the thing I just described is impossible? And that's part of the question.
Understood. So you're focusing on development of the toolkit.
Yeah, at this stage, we're focusing on developing the toolkit, as well as, I mean, that comes with a bunch of scientific discovery because it's very poorly understood. So I think that's a goal in and of itself too, is to understand how does this organism generate force. And I would say that that's also a less synthetic cell focused rule of life, but also in terms of the broader scope of the rules of life. What are rules of life that govern mechanical force generation? I mean, we have sort of thought of cellular force generation as being dependent on consuming ATP, and this is an exception to that rule for sure. And what other rules might it be exceptional to?
Mary, I have a question. So there's kind of this big discussion in the community about building cytoskeleton for cell division. Is that something you guys thought about?
We have thought about that some. I mean, I think like our sort of initial goal has been more tool focused, but I think that's certainly one application that you can imagine. And many members of our team, sort of our main projects are working on cytoskeletal machinery involved in cell division. But you can imagine, for example, once we have this in vitro reconstituted protein system, if you could anchor it around the outside of the cell membrane, you can imagine that contraction being useful for, you know, something like a contractile ring. And I think for sort of the chromosome segregation part of cell division, I think it's probably less easy to see how this particular tool would be applied to that. The other thing I mentioned that we've developed was this magnetic tweezers approach to moving things around inside cells, and I think that might be more productive for sort of delivering two things to the right place. Like that needs a larger sort of director to say, okay, where are the two ends of the cell and where am I going to, as opposed to the overall contraction that you want when you're segregating the cytoplasm. So I think those are still, like, you know, long-term goals, but yes, we are thinking about how, you know, how these things might be useful for controlling cell division. And I think there's a real potential power to using machines that are independent of the cell types that we usually use, so that you can manipulate them without disrupting, you know, the things that are already there in the cell.
Can I ask a very quick question, Mary? So I'm curious, have you guys been able to do this in vitro yet? And what's the concentration that's needed to do this contraction? I'm just thinking if you were to put it in
The
a
concentration
cell,
of protein or of
calcium level that's required to
I don't have that number off the top of my head. I'm sorry. I mean, the answer is yes, we can generate contraction in vitro with purified proteins. And in response to, you know, changing calcium, for example, either by, you know, pipetting in calcium or by light activated release of caged calcium. But I don't know that concentration off the top of my head.
Yeah, but just thinking, if you were to put in a cell that you know, calcium is utilized in many processes.
Yeah, no, I mean, right. So I think that is a potential cell. I mean, it's very local. So if you can manipulate it locally, I think you can imagine that maybe you wouldn't break everything. But yes, I agree that's a potential issue with the approach on that one. But you can imagine thinking about, like, other ways to, you know, to regulate it. But yes, I agree that that's a potential challenge.
Thank you.
Vincent, I saw your hand, but maybe you were going to say the same thing.
Yeah, it was more or less the same thing.
Yeah.
Apparently, you have identified the main components. That's it. Of this mechanism.
Oh, the contraction. Yeah, no, we think we have. Yeah.
Oh, okay.
I mean, that. Yeah. I mean, I would say that's somewhat silver. That part's not published yet, but I would say I think that we have.
And are there any sort of analogies with, for example, myosin and actin, or kinesin and microtubules?
So I would say that it's still early days in terms of mechanism at that level. I mean, I think, yeah, and I don't think we know yet. Yeah, okay. It's probably the best answer. I think that it's quite different, but I mean, I also think we don't know for sure. But I would say that the initial signs are that it's really quite different in terms of how this machinery contracts. I mean, it may even be more like a phase separation. I don't know. That's speculative, and maybe I shouldn't say that, but that's one of our speculations at this point.
Just one quick comment for the PIs. It would be very useful if perhaps you could punctuate and summarize perhaps the rule of life that you are developing or have discovered.
Okay, I mean, I would say on that, the rule of life, I think we're understanding is how can cells generate mechanical force. That's across evolution, and how many different ways have cells evolved to generate mechanical force.
Alan, can you do the same thing?
Yeah, I was hoping that I don't have to do that, but that's all right. I would say the rule that we hope to identify is that the propagation electrical information are just purely mediated by the ion channels, and nothing else is required. I think that would be a rule would love to be able to bolster. What I also forgot to mention is that we have tried doing this in living cells and taking a non-electrically active cell like a HEG cell and putting in channels to show that it can be electrically active. Some people have done that before, but we're also trying to repeat some of those studies and actually changing different types of voltage gated channel, and you actually see different spiking activities, which is very neat to and that's I guess another lesson there that learned there is it's probably the ratios of the ion channels that matters in terms of shaping spikes rather than the absolute concentrations of ion channels. So,
okay, thanks. I think Farron, you're up next.
Do you want to know my role of life too?
Yes.
So I would say that we discovered, much to our chagrin, a rule of life, which is if you want to make Narnavirus into an RNA chromosome, forget about it. And the second one, and the serious one, is
Jeff, that's already a well-known rule of life. You can't fool us.
and the second one is that this very unusual class of tiny viruses seems to depend on pervasive RNA folding from the first base to the last.
Okay,
which
thanks.
we sure didn't know.
All right, Farron.
Okay, thanks everyone. So as a reminder, I'll give a synopsis of the project we fondly refer to as booting up a mirror cell. So the long-term goal and vision is to design and construct synthetic mirror cells that are comprised of the key molecules like nucleic acids, proteins that exist in their the opposite chiral states. So rather than a D nucleic acid, an L nucleic acid, rather than an L amino acid, a D amino acid. And so that kind of goes back to some big questions that you guys were asking us to put out there. It's really serves as inspiration to see if we can really challenge some of the big ideas behind the central dogma, right? Can we actually manipulate the natural biomolecular machines, polymerases, translation machinery, ribosomes that can actually polymerize and accommodate an entire mirror system that can coexist almost orthogonally from the natural living world. And in doing so, we'll gain obviously a much more deeper fundamental understanding of the role of chiral structures in biological systems, particularly the role of chirality, and to what constraints existing machinery and enzymes might have on the productions of these molecules. So when you think about, let's say, the role of chirality, you can think about it playing a big role in the folding interactions of different biomolecules. It can certainly drive differences in crowding and aggregation, the formation of organelles, as well as playing a big role in structuring and patterning, and playing also a tremendous role in development as well. And we also have some interesting applications in mind where you could imagine, for example, producing new types of D proteins or D peptides could set the stage for the development of new kinds of drugs, for example, that might be immune to proteases or nucleases that are naturally in the body. I'll now kind of walk you through some highlights of some efforts that our group has been pursuing, and they broadly have sort of encompassed an effort on sort of chemical synthesis of D proteins and peptides. That work has largely been out of the lab of Neil Devaraj, where he's developed a new kind of a method referred to as lipid-facilitated native chemical ligation. It's basically a method that allows the formation of peptide bonds rather at the ligation site. And this basically has opened up lots of avenues in terms of accelerating reactions and expanding the repertoire of chemical ligation sites. He's further developed this to basically use split intine-mediated ligations for the synthesis of transmembrane proteins, and he's basically adapting these strategies to be able to chemically synthesize large stretches of peptides and proteins comprised exclusively of D amino acids. In some complementary work, Irene Shen's been asking questions regarding constraints of chiral recognition of RNA elements. And in particular, she's been looking at RNA ribozymes that allow the self-aminoacylating of ribozymes. And she's already proved this out a number of years ago, but in the context of this project, she was looking at whether there are specific preferences or discriminations, for example, against the aminoacylation of D amino acids compared to L amino acids. And surprisingly, in some of her in vitro experiments, she finds that there is not a strong preference for L over D. In some complementary work, other folks in our group have been trying to really engineer biomolecular machinery like polymerases or components of the translation apparatus to basically polymerize and encode D amino acids at the ribosome. And so this can be broken down into various, sub-projects where you can think about engineering certain types of synthetases that can recognize and charge tRNAs that can stably hold a D amino acid that could then be delivered to a ribosome. And then the question then becomes, can it get past some of the EFTU trafficking? Does the catalytic core of the ribosome accommodate a D amino acid and allow amide bond formation with D amino acids or strings or combinations of LMDs? Along these lines, our group has identified some proof of concept where we can actually enhance the and delivery of D amino acids into cells. We show evidence of being able to charge tRNAs with D amino acids inside cells. And we've also developed abilities to drive molecular evolution of a special class of engineered ribosomes, tethered ribosomes, that can be separated from the natural ribosomes. And in the context of this project, are being engineered to accommodate D amino acids. And so in doing so, we've basically developed capabilities to engineer the entire ribosome, including the peptidyl transferase center, and have also developed basically an in vivo fitness landscape of this ribosome and have shown how surprisingly malleable it be. And so that's kind of a summary of I think a lot of our technical progress. Like some of the other projects, there's been a lot of advances on some of the tools and technologies towards this bigger vision of booting up a mirror cell, which is why we termed the project Booting Up. But we do think that we've made some important steps along the way, and we're still actively driving this research forward and excited about continuing it even beyond this current project. Happy to take any questions.
Yeah, I have a really quick question. Very interesting. So nested in all of this is an important origins question. So why does life choose L over D? And you have any maybe words or insights based on what you have experienced in your research that would perhaps facilitate an answer to that question?
Yeah, that's a really good question. I don't think it's one that I have a clear answer for. But obviously it's conserved across all life forms. I think what's really interesting that kind of builds off of that is you have, D-nucleic acids that are interacting with L amino acids. You've got sort of this complementary transition between D and L molecular forms. I don't know that we necessarily have elucidated, a great answer to that sort of fundamental question. And in fact, we do see instances where you lose discrimination, as in the case of Irene Chen's work. So there is some sort of flexibility or malleability in these molecular structures, but clearly with lots of other, biochemical reactions or just interactions between biomolecules, having a conserved defined chiral structure obviously has been critical in evolution and maintenance of biological function. I'd open up to Kate, who's also been thinking about this too, to see if you have anything to add, who's also part of our team.
I think Farron really well described really well what we were doing and answered the question. The one thing that I'm really excited about this project is how generalizable that technology is. We're using in vitro ribosome evolution, kind of complementing what Farron is doing in cells. We're doing it in a completely dead, pure system. And we're basically learning all the skills and technologies we need, not only for the opposite chirality building blocks, but also for all different kinds of unnatural monomers, including different kinds of amino acid chemistries. And that kind of emphasizes the need to evolve parts of the ribosome like exit tunnel and the PTC that we're evolving in vitro right now. And obviously I'm looking at the origins implications too. And it's kind of more of a platform technology development at this point, even though we're not supposed to develop platforms anymore.
So Farron, are you able to get an amide bond between two D-amino acids in an evolved ribosome?
Not yet in a cell, right? As you know, there's literature from years ago that has shown this in vitro, but we have not achieved that goal in a cell yet. We're working towards that.
So do you have a sense of, at some point in the origins of life, wherever the major steps of proofreading were to enhance chirality one way or another, if they took place outside of the ribosome, it would be pretty easy to manipulate the ribosome to accept D-amino acids. Do you have a sense of where that proofreading would have taken place? I know some of it must be in the ribosome, but where the majority of that, if Irene's making synthetases that easily charge D-amino acids onto tRNAs, and if EFTU has to be engineered, then that would suggest that the ribosome should be amenable towards D-amino acids, maybe. Just some thoughts on that.
Yeah, sure. That's really good questions. So the short answer is that there are multiple areas where there are sort of safeguards that prevent the encoding of D-amino acids at the ribosome. So there are proteases that are hanging around in the cytoplasm that will degrade both free-floating D-amino acids, or in some cases, those that might have been inadvertently charged onto a tRNA. And so part of our efforts are identifying those, creating strains where those are knocked out. And in doing so, we actually do see increases in the ability to sustain D-amino acids as well as charged tRNAs. Then you're right. The next step is, to what extent is that going to be compatible with EFTU? We've done a little bit of engineering in EFTU, but not a huge amount. We then shifted a lot of the focus to the ribosome, where you could imagine the catalytic core is going to be sterically constrained and evolve to really just accommodate your alpha amino acid chemistry. There have been some modifications of ribosomes by Virginia Cornish and Ruben Gonzalez, for example, that have showed engineered ribosomes that can better accommodate the amino acids in vitro. And what we've actually done is introduced these orthogonal tethered ribosomes and have evolved those and have started to select those that can preferentially accommodate D amino acids over L. So the way to think about it is, you have a really good question. There's probably multiple layers of safeguards that are present in natural cells that protect against the encoding of the mirror image D amino acids. And we really have to approach the problem from all of those dimensions simultaneously, and then try to put them together as a single system and do sort of systems engineering and optimization once we start to work out the role of each of those individually. Hopefully that gives you a appreciation for the challenges and the approach that we have to take.
Yeah, certainly. So if you were a betting person, you're taking a multi-pronged approach. You're trying to do it chemically to make peptides that have D amino acids, and you're trying to manipulate the ribosome and the translation components. What do you think is going to win?
Well, today, the chemical methods have clearly won, right? They've been very successful at producing D proteins. So I don't know if one winning or not. I think it will certainly be if we can pull off, or if someone out there can encode D amino acids inside cells. I think that would be an impressive feat. Being able to polymerize entire proteins for all, let's say, 20 D amino acids is a daunting task. We're years away from that, but we want to try to at least establish feasibility.
Okay, so before we move on, back to Corey's point. So what's your rule that you have discovered or that you've gotten insight into?
Well, I think a few things. One, that the machinery that's responsible for producing these molecules actually is quite flexible. And so between that and I think a lot of proof of concept and cell-free efforts, I think it's establishing a path towards a mirror cell. And I think there's a lot of potential, as Kate was saying, from an applied perspective, to go beyond just what we're talking about in this context and really opening up a new possibility of repurposing ribosomes to create entirely new kinds of polymers and materials.
Okay, cool. Jeff, you have a question?
Yeah, I was just wondering, if I remember right, there's a class of antibiotics like valinomycin, which contain multiple D amino acid linkages, and they're not made on ribosomes. But is there anything to learn from the cells that produce those molecules? Because they obviously are not degrading D amino acids, and furthermore, building useful things out of them. So any comments?
Yeah, that's a good question. Yes, most that's made through NRPS-based mechanisms. And what we've seen is that the enzymes that recognize and degrade the D amino acids are obviously going to have high specificity. So it's very likely that in many of these cases, you don't have the certain proteases that might degrade the certain types of amino acids that might exist in those antibiotics, or in the periplasm, there's also instances of D amino acids. And so if you look across actually diversity of organisms, you do see different types of proteases specific for different types of D amino acids. And I think that opens up avenues to produce stable D amino acids. And I think as we start to better map those, we can create engineered strains that lack those types of proteases, which is what we're doing in the strains that we're working on.
Thank you very much. If there are no more questions to Farhan, let's move on so we don't run out of time going through all of them. So let's quickly go back to Mary about the question that was asked from the audience.
Yeah, so there was a question about, do you need cytoskeleton for genomic division, and about what's used for genome replication in synthetic systems already. So I think this question, if I understand it right, there's sort of two parts of this question. How do you replicate a genome synthetically, and also how do you segregate it? And so whether you need a cytoskeleton for division, I think depends how much you care about copy number, right? So if you can make a whole bunch of copies of something, then you don't necessarily need anything to segregate it correctly, because you can have many copies randomly distribute themselves. But if you want to have exactly one copy, which is usually the case with a sort of whole genome division in a higher eukaryotic cell where you have a chromosome, then you need something to generate force to deliver it to the two halves of the cell. And I think I would call the thing that does that a cytoskeleton. There's lots of different ways that cells can do that. And I think that's a separate question from replication. Yeah, hopefully I answered the question. Jeff, I don't know if you wanted to further weigh in.
Oh yeah, I was just saying that the specific question was whether you can substitute cell genome replication systems with phage polymerases, and to my knowledge, that's not possible. For exactly the reasons you said, that if you want control of one replication per cell cycle and copy number control, you need a lot of machinery.
Vincent, go ahead.
Yeah, I wanted to add a comment complementary to Mary, what Mary has said about this question from Sean Hamilton. Do you need cytoskeleton from genome for genome division? I'd like to point out just the work, and I don't know precisely if the answer is there, but beautiful work by Jeff Harrington. It's now 10 years old, 2013, published in Cell. The title is Excess Membrane Synthesis Drives a Primitive Mode of Cell Proliferation. And they showed that you don't need FTSZ, and apparently you don't need any cytoskeleton to proliferate a bacterium, just by excess membrane synthesis. It was a very interesting work, and a possible mechanism, primitive mechanism of how our cells can expand, divide just by excess membrane synthesis. That's it.
Yeah, no, I think that's a really good point. I lied that there's certainly bacterial systems that don't have cytoskeleton for dividing their genomes, but then you end up with multiple copies of the,
exactly.
Yeah, exactly. Like, right. Yeah. Yes, exactly. Yeah.
It's a little bit messy, but it works apparently.
Thank you so much. All right, so let's move on to Cindy.
Okay, great. Can you hear me better now?
Yes, we can. Thank you.
Okay. All right. So yeah, I just wanna just repeat that our long-term goal for project is to study wound healing mechanism in this giant single-cell organism called Stantrus aureus. It's free living. It's a ciliate that you can find in quite a lot of freshwater ponds, and it's been long known that you can cut the cell into many small pieces, and as long as each piece has some DNA, it can regenerate in full. And my collaborator, Professor Wallace Marshall at UCSF, has been studying that mechanism. But one of the maybe less known, I guess, mechanism is when you cut the cell, inevitably you're going to introduce wounds. So the question is, how can the cell survive through such drastic wounds when you cut it into many pieces to start with. So that's sort of the goal of the our overall project, which is to understand the underlying repair mechanism of this giant single cell. Now, in terms of the project, took us some time to build the right tools to introduce precise cuts in the cell, which is more challenging than what you might think, because the cell is very motile. So again, it's a cilia, so it has very strong cilia, so it can swim around. It's a strong swimmer. So finally, we're able to build the right tools, and we can control the wound size precisely. So now we have characterized the wound healing capacity of the cell. We know roughly what's the wound healing rate, and also what is the maximum wound size that these cells can heal. And what is really surprising, maybe not that surprising, given that it can regenerate so well, is that it can heal membrane wounds that are over half of the entire cell surface area. So what basically more than half of the cell surface will be gone, and the cell can still survive and heal and regenerate subsequently. And some of the cells, we found that they can heal up to 80% of the membrane wound. And the time scale is on the order of 5 to 10 minutes when the cell can close its wound. So now that we have some understanding of what the cell can do, of course, there are many questions about exactly how it does it, right? So of course, we're interested in how the cell can heal drastic wounds, but also just a simple question: how can the cell survive such a large open wound for so long, or even if it does nothing, how can it be able to tolerate any influx of external calcium or toxic chemicals or ROS radicals from outside in an open wound that lasts for 5 to 10 minutes. And then, of course, finally, we're very interested in extracting any principles that we can find from this cell to apply possibly to the engineering of synthetic cells to make them more robust, to have this wound healing capacity in synthetic cells. So in terms of the discovery, I think maybe the most surprising thing that we found so far is that at the wound site, of course, there are events that have been described before in other wound healing models, that there are vesicle transport, but basically lipids that are transported from the inside of the cell to the wound site to patch the wound. But in addition to that, we found that the cell can use these really large-scale mechanical motions like folding and contraction and twisting and pulling and swimming to physically reduce the wound size first. That happens basically immediately after the wound is introduced. So we're still in the middle of understanding what drives these mechanical motions or mechanical modes of wound response. But I think that could be the rule of life moment where these large-scale mechanical behaviors may be of greater importance than we thought, and especially in single cell wound repairs, especially for single cells that are free living and also might have ways to swim around like these cilias. And we think that this could potentially be applicable to the thinking of the design of synthetic cells, since a lot of people have already been trying to incorporate cilia and other ways of introducing mechanical motion in synthetic cells.
Just a little housekeeping. We have 3-ish vignettes, so we're probably going to have to speed up just a little bit. With that said,
but this is the best part, Corey.
Yeah, it is. It is the best part. I just, yeah, we can.
I'll be cognizant of time. Okay. So Cindy, I have a question about your research. Are the liposomes just delivering materials, or are the liposomes themselves being integrated into the new membrane?
Yeah, we think that they are integrated into the new membrane, and then of course over longer term, time scale, eventually they probably get recycled and maybe endocytosed, and then they get kind of recycled within the cell to restore the original composition of the plasma membrane. But that's something that we're very interested in looking at as well.
Okay, so if harping back to Corey's point, can you make progress in generating a rule for life related to this research?
Yeah, so I mentioned so that this the most surprising finding was that the is using these mechanical behaviors. So literally the cell folds on itself around the wound to basically reduce the wound size physically. And there are other motions too that we think that might happen also in other cell types that are able to swim and also change the cell shape freely. So this might be the rule of life that like a lot of people have missed, especially in the wound healing literature, that people just like, oh, it's like biochemical, reaction that you patch a hole with lipids, but there's nothing that's like macro scale involving the whole cell that folds around the wound.
Okay, thanks.
So I have a quick question. How many nuclei does one stentor have? And how many fragments can regenerate a living organism?
Yeah, so stentor has many micronuclear nodes, I think on the order of like 10 to 12, depending on, like feeding conditions, and basically many copies of it. So that's why you can, cut into small pieces, and as long as you some, you can regenerate. The smallest that can regenerate, that was actually found first reported in the 1900s, early 1900s, was about 1/27th of the original cell size. And of course, we're very interested in asking what is special about that size, right? So that's something that we're also trying to reproduce and see if we can really get make sense of that number that's been reported in the literature since the early days.
Cindy, I have a question. So you talked about sort of the cell wanting to protect itself from bad things from outside getting in. What about things leaking out? Do you think which of those does it care more about, I guess?
Yeah, absolutely. So the cell, that definitely lose quite a lot of the cytoplasm once there's an open wound, and we're also characterizing how much is it losing, and we found that it definitely lose at least 20% of the cytoplasm. That's what we have observed at least for the experiment and the conditions that we're testing. But we say that maybe that the cell is okay because it can, like, obviously we can cut it into many small pieces, and each piece has less material of the original cell, right? So I think if the cell lose some cytoplasm, maybe it doesn't care as much as long as some essential components. So that's another question we're interested in. So what is the minimum set of components that are needed for the cell to be able to heal and regenerate?
Thanks.
Okay, if we don't have any more questions, we can move on to Chris.
Great, thank you. So the goals of our project were, like other projects here, to build a synthetic cell. And I'll mostly talk about some of the theoretical work that we did within the context of that project. So I think when we think about the diversity of life, especially the diversity of bacteria, we often think at two ends of a spectrum. One end, we say sort of everything is different. Every bacterial species is unique. It's evolved to a particular niche. It has a particular set of metabolic pathways and a particular genomic composition. And we think about these species as being very diverse from each other, and sort of in many cases for prokaryotes being overwhelmed by that diversity. And then at the other end of the spectrum, we often think that sort of everything's the same. And what I mean by that is that all of the cells share the same dominant macromolecules. So from a macromolecule perspective, we often think of bacterial cells as sort of all being one thing. And so I think for thinking about designing synthetic cells and bioengineering more generally, we need theories and descriptions that sort of live in between those two ends of the spectrum. So something that shows us what's the same about bacteria, but still tells us what the key dimensions of trade-offs or differences are across that diversity. And so one way to look at this is through the lens of scaling laws. And so when I say a scaling law, what I really mean is that if we look at, say, the size of a cell, what's shifting in its energetics, physiology, elemental composition, so forth, sort of anything that we might really care about for cells. And what we found is that there are really strong scaling relationships relating metabolic rate, dominant macromolecular composition, genomic composition, and even elemental abundances, relating all of those features to the cell size of an organism. And so a scaling law is really just cell size to some power, to some exponent. And so we find lots of these relationships sort of for all of the key physiological and metabolic processes that we'd be interested in. Often we have theory that can predict where these scaling laws come from, can predict what the value of those exponents should be. And often what you're seeing there is that organisms are evolving to hard and fast physical constraints over evolutionary history. And so really it's sort of physics predicting what you should see in the physiology. And so what's nice is that that's kind of a regularity or a law that goes across all of bacterial diversity. But it's also a set of laws that tells you how radically different large cells are from small cells. So as an example, in the smallest bacterial cells, one would predict that, or one also observes in the data, but also would predict from theory that the cell is sort of mostly DNA and expressed proteins with a handful of the RNA components like ribosomes and tRNA and mRNA. And so those cells are dominated by the functional proteins and the genomic information to actually store what the cell is and does. And then as cells get bigger, you rapidly dilute out the protein concentration and the DNA concentration, and all of the RNA components become much more abundant as cells get larger. And so this tells you that if you're building a small cell, you need to build it a fundamentally different way than if you're building a large cell, because the ratios of macromolecules are dramatically changing, and the overall concentrations are dramatically changing. And then we've been able to show in this project that has downstream consequences on stoichiometry. So small cells are very nitrogen dominated, large cells are much more, small cells are much more phosphorus dominated, large cells are much more nitrogen dominated. So that tells you something about what these organisms require from their environment, and how you might have to design environments for different types of synthetic cells. And then we've also seen that you can take this all the way to sort of detailed information about what's stored in the genome. So if we look at enzyme categories like ligases and so forth, all of those have strong scaling relationships with genome size. And that was work done with Sarah Walker and her group at ASU. And so sort of putting all of that together, we start to get a picture of what types of trade-offs you need to have in building a synthetic cell. And what it does is it takes this huge phase space, this huge sort of trade-off space of physiological possibilities, and for any size of cell puts you in a much narrower region where you sort of know what to start from in building a synthetic cell at that scale. And the next step is to actually implement those different design trade-offs and see how well they work when you're building radically different types of cells. And so I also want to mention that for me, the rule that emerges here is really looking for scaling laws in life, often because life is evolving to physical constraints, and physical constraints naturally come with scaling laws. And I'll mention that we see these same sorts of scaling laws, not with the same exponents, but the fact that you do see scaling is something that shows up in unicellular eukaryotes, something that shows up in metazoans. We see them for large vascular plants. In each of those cases, we often have good theory explaining why certain physiological features are scaling in the way that they do with the overall size of an organism. And so I think that's a very general set of concepts that sort of cuts across the tree of life, and in this case is very useful for designing small synthetic cells. So that's all I'll say, unless there are questions, and thank you for your time.
So Chris, I had a quick question with regard to, so you mentioned a lot of stuff about composition, but how much of this is limited by transport phenomena?
Yeah. So we did some work on that here as well. So you can write down nice optimization processes for either how many transporters you need on the surface of the cell to facilitate that sort of transport. And then we've also done some work on sort of internal transport constraints. And so putting those together, you also get nice scaling relationships. But you can predict, for example, at different cell sizes where you saturate total uptake based on the number of transporters. And so we showed that as an example in a pretty general paper about these different types of trade-offs. And so, yeah, there's a point at which adding transporters, you incur a cost for building them and you don't really get any return on uptake rates. And that's very important. And that's also very scale dependent, and has sort of strong asymptotic behavior at the small end. And that's also something we see in a lot of these laws, is that usually at the smaller large end, you have sort of strong asymptotes that make a certain architecture very difficult to continue along. And we think that's what motivates major evolutionary transitions, is just a certain architecture starts to break down. So for the example, in the smallest bacteria, you start to want to cover the whole cell and transporters, and that's an issue in terms of having structural integrity and space for other things.
Any other questions for Chris? If there are none, I just want to say, Chris, this is awesome project because it kind of ties in a lot of the projects in this space. You're basically summarizing the rules of life that we're finding during the experiments.
Yeah, ever more to discover, but yeah, thank you.
Thank you so much. Okay, so let's move on to Vincent.
All right, so the ProteoCell project was a fat-free cell project. So what we did is that we look at the living cells, as we know, and they are composed of 4 types of biomolecules: the carbohydrates, the lipids, the nucleic acids, and the proteins. And we came up with this idea, with this question: can we make a synthetic cell from the ground up? We are going to take molecules without one of these types. We are going to remove one. And so we look at those 4 types. We had some discussions, the state of the art of bottom-up synthetic cells, the skills of the group, and we decided to build a synthetic cell without lipids, and especially without phospholipids. And the phospholipids, they make the physical boundary of cells, and we decided to try to make a cell without this. And so we relatively quickly converged towards liquid-liquid phase separation. When you put some polymers in an aqueous solution, spontaneously, many polymers, they are going to form droplets without membrane. That's it. And we have worked on this during this NSF-sponsored project. And there was different tracks in parallel, and one of them was to do this in a buffer. So we take polymers and then we make droplets. It's relatively well established, and Christine Keating is a very popular, very famous researcher in this field. Yeah. And the polymers are PEG, dextran, ficol. They are all biocompatible polymers. And when you put this in a buffer, they spontaneously make droplets, cell-sized droplets from 1 micrometer to 30, 40 micrometers. And what we have done, the group of Cheryl Kerfeld, they purified some proteins that are known to make compartments. They come from BMCs, bacterial microcompartments, but they are much smaller, 100 nanometers. And they make some sort of capsids, very well shaped, and they make compartment. And the group of Cheryl, they purified many of those proteins, and then they sent that to the group of Christine. And when we mix those proteins with those liquid-liquid phase separation, these proteins spontaneously, they go at the interface. They kind of almost replace a phospholipid bilayer. And several proteins have been tested. They are fluorescently labeled. We can see them, and really spontaneously they go at the interface. We were not able until now. We are not able to do, for example, some electron microscopy to see what are the structure at the surface. But at least they go at the interface, and with that, we are trying to replace the phospholipid with proteins. We don't want lipids, we remove them, and we are trying to make a membrane made of proteins. And so we achieve that, and my group, with collaboration with other groups, Millicent Sullivan, and then Giovanna, we are trying to use cell-free gene expression. Alan was talking about cell-free gene expression for his project. And we are trying now to make liquid-liquid phase separation in the cell-free gene expression system and try to use those proteins used by Cheryl and Christine to express them and put them at the interface. That's it. So that now we have synthetic cells with a membrane with these proteins, and we have gene expression inside like a cell. And we start seeing something. We were able to make liquid phase separation in a cell-free gene expression system. It's a relatively complex solution, but we are able now to do it. And we have expressed some of those proteins, and we see co-localization. And we have beautiful images on this. So the rule of life for this project is that we do not rule out the possibility of making synthetic cell without one of the 4 types of biomolecule, which is the lipids. So far, it seems achievable, and we absolutely don't see any major issue, any experimental evidence that it's impossible, that's not going to work. So far, it's absolutely plausible, possible. That's it.
Alan, go ahead.
Well, yeah, sure. Very interesting. Let me put my hands. Vincent. I was curious if you can comment about like what other type of proteins might go to the interface. I know you said you work with the bacterial microcompartment proteins. Do other capsid-like proteins also can go to the compartment?
Very good point. Yeah, that's a great point. So far, we tested BMCs. Some of them, they go at the interface very nicely. Capsid proteins from bacteriophage are in the list of what we want to try, absolutely. And what I tried is the MREB protein from E. coli in my lab because we had some experience with it. MREB, it's a cytoskeletal protein in E. coli that makes E. coli a cylinder. It polymerizes at the membrane. It interacts with the membrane. It's not a membrane protein. It's a cytoskeletal protein that interacts at the surface, at the inner membrane. That's it. And when you express MREB, it co-localizes spontaneously at the interface of the droplets. It's really cool. That's it. Yeah.
So other proteins also have that similar behavior. Yeah, I was wondering if you could figure out the rules of life of what type of proteins might actually be at the interface versus, in the lumen of the phase separated droplets.
That's a good point. We are testing different proteins, and maybe at some point we could say something about this. Absolutely. Yeah.
Very cool. Thanks.
All right, thanks, Kate. I think that wraps up the scientific achievements portion.
Eric,
yep.
Just one more thing. We have a question from John to Chris, so maybe we can answer that before we move on.
Okay.
Yeah, thanks. So John's question, so this is from John Glass, who was the PI on the project that I was on. Our minimal cell that lacks FDSZ and other cell division machinery is vastly larger, 100-fold more volume than cells that undergo normal cell division where the septum forms between daughter cells. How does that fit your model? So I think this is a great point. So in a lot of natural populations, cell size is a good proxy for all of these other constraints because the cells have had a long time to co-adapt all these physiological features together in an environment. And so cell size is often the dominant variable we use against which to measure everything else. I think in more general context, though, what we really want to think about is taking the interconnected scaling between all sorts of pairwise physiological features, say growth rate and protein concentration, and ribosomal abundance, where we can often relate all of those to cell size, but you can also do sort of all of the pairwise scaling relationships and get this high dimensional space of trade-offs. And then if you have a good model that predicts all of those interconnections, you sort of understand why all the pairwise scalings are what they should be. And then for any sort of either particular species that's adapted to a bizarre environment or any novelly produced organism, say a synthetic organism or highly engineered organism, what you can do is just say it takes maybe it takes a big step in one dimension of that feature space. What else comes along for the ride or what else doesn't change? So for example, you might have a big shift in volume without a large change in growth rate. And if growth rate doesn't change, then you'd predict a bunch of other things from growth rate rather than cell volume. And so you can sort of then start to think about it as this, high dimensional space of co-constraints and asking where are the sort of rigid dimensions and where are the flexible dimensions for these sorts of perturbations. I think that's the next level of sort of more complicated theoretical thinking, and addresses John's question. So yeah, thanks for that, John.
Thank you so much, Chris. Okay, so now we wrapped up the science part, and Eric, back to you.
All right, thanks, Kate. So for time reasons, I think we can get away with about 20 minutes in the next section. And the next section is going to be two components. One is multidisciplinarity, and the other is training. And since we have, I guess, 7 of you, I really want to limit your responses to about 2 to 3 minutes. And I would like each of you to answer 2 questions. One question is, what has been challenging about creating or maintaining your multidisciplinary team? And the other question is, what challenges or opportunities have you faced regarding the training of students given the multidisciplinary nature of your teams? And so I'm just going to go down the list of participants that we have. So Alan, I have you listed first. So if you could just address those 2 aspects, and if you want me to repeat them, I'll be happy to.
Yeah.
Just one quick thing before you start on. We can go until 2:20, so you have about 30 minutes, so a little bit more time.
I'll still keep it to 2 to 3 minutes. So yeah, but maybe there would be some back and forth after. So first was about challenges in multidisciplinary team. I think that was the first question. Is that right, Eric?
Yeah, correct.
So we've been running, maybe just to step back a little bit. We've been running this project for close to 4 years now. We've been, having a monthly Zoom meeting with all team members, and then in between we use a Slack, tool to help conversations in between the monthly meetings. It's worked well, and we basically rotate between labs to present. I think the challenge would have been, I guess, maybe let me phrase it this way. So we have projects both on the very bottom up and slightly from the sort of the middle ground level where we're engineering cells. And I think it's very difficult to envision a project that really merges the 2 together. We try a little bit, and we had one paper last year where we had a little bit of complete bottom up approach plus some of the cell-based work. But in reality is merging these 2 sort of system is not easy. And that, I think that presents a challenge. But on the other hand, in terms of training, I think the second part is opportunity and challenges for training. I think the students do learn a lot about how different science works, and I think this is one of the goal of this Rules of Light program is to train interdisciplinary, multidisciplinary scientists. So we have people from physics, people from engineering, people from the life sciences, and we sit through all these meetings, and we have the social science as well. I personally have learned a great deal about thinking about how synthetic cells might benefit society, and these are the questions I normally don't think about day to day, but the social scientist is able to bring in a very sort of different vantage point that I think all the trainees get to hear and learn about. The opportunities have been great. We have one in-person meeting at the very beginning, and then COVID basically stopped that interactions. We are hoping to have some lab visits later on this year to still facilitate that in-person interaction. So maybe I'll just leave it at that.
Yeah, that was perfect. Thank you. All right, Mary, we go next, please.
Sure. So I would say a lot of our strategies kind of overlap with what Alan just said in terms of structure. So I'll kind of skip those things and just focus on things that are maybe a little bit different. So the biggest challenge I think was that our team started, just at the beginning of the pandemic. And so while the PIs had really gotten to know each other at the NSF Ideas Lab meeting, the trainees had less opportunity to do that before. We actually had not had an in-person gathering before COVID. And I think the biggest thing we used to combat that was really a lot of the skills that we learned in terms of generating shared language that we learned from the Ideas Lab. So I actually felt like the activities at the Ideas Lab were really helpful skills that the PIs took on and then passed on to our trainees. And then the social scientists helped kind of on our team came up with sort of a set of questions to help us keep focused on the big picture, which was the way to kind of keep us from getting bogged down on these ideas, kind of like Ellen was saying, of how do we push all this project together? It doesn't necessarily have to, but we want to, stand back and think about the big picture. And then I think the opportunity to get together in person and do experiments together at the Marine Biological Laboratory in Woods Hole last summer finally was really key, especially for the trainees to have them sort of all be in the same physical space together. The other challenge on the trainee side, I would say, is in recruiting onto an interdisciplinary project, right? Especially starting from scratch. But I think I do think it, as Alan said, it poses a great opportunity for the students to really learn how to talk across disciplinary boundaries, which is a really key skill that's useful, no matter what they do next.
Yeah. So were your trainees present? I just assumed in Alan's meetings, his trainees were presenting. Was that the case for?
Oh yeah, sure. So yeah, our trainees were also presenting for the most part. I mean, sometimes the PIs would present, but for the most part, the trainees would present. And so sort of like one at a time, they would get to know each other's projects. But because our team is so diverse, it took a little while to get to where we all had shared language to talk about it. And I think an in-person retreat for a couple days probably would have gone a long way toward helping with that. But unfortunately, the timing was not in our favor for that.
Yeah. Okay, great. Thanks, Mary. Farron, you're up next.
Thanks. Yeah, I would probably echo what we just heard. Our team is really comprised of people with expertise across chemistry, biochemistry, molecular biology, and bioengineering primarily, and also some computational biology. So there was a lot of opportunity for students and postdocs to get exposure to different fields. I would certainly say that given the fact that the pandemic overlapped with most of the project, it did create a barrier to drive lab visits, which I think are really critical in overcoming some barriers across disciplines, or to help learn certain types of techniques. But we've did our best through lots of more targeted like subgroup meetings and collaborative efforts on sub projects to really try to share resources and knowledge. We are actually hoping to schedule some in-person meeting to get our teams together, with the hope of actually using that to then submit more collaborative grants in the future. With regard to other challenges with the students, I would actually say they typically did a really good job. I think they took a lot of initiative, reaching out to other students or labs. I would say, yeah, the missing piece was not being able to physically visit other labs for training, which obviously was just unique to the recent circumstances.
So Farron, I didn't catch everyone on your project. Do you have formal engineers on your project?
I'm trained as an engineer, so yes.
Yeah, but your position is not an engineering position. I mean, do you have engineering faculty?
I am in the biomedical engineering department.
You are?
Yep.
I didn't know that. I thought you were just.
I'm in two departments. That's one of them.
Okay. So you get students from engineering, and you get science students.
Mm-hmm.
At the end of the day, meaning at the end of their PhD, is there a lot of difference in those two students?
Yeah, that's a really good question. So those students will undergo different formal training and coursework and qualifying exams. So their formal training is different. And to your other sort of larger point, the projects that they pursue will largely govern the skills that they develop. And, you know, a lot of the projects do cross disciplines. But most of the differences are going to be rooted in the formal training that they have in their different programs. But most students that graduate my lab will have both experimental and computational skills.
So would you say that at least for the molecular sciences, there's less of a dichotomy between engineering and science than there used to be, or how things were done in the past?
I think there's still like fundamental differences in just approaches to questions and projects. But I think there's more, I think, fusion between those areas. And also I think there's more efforts even I'll just speak to Yale at an institutional level to forge more interactions and collaborations between the biological sciences and engineering. Both through coursework and through collaborations and joint mentorships.
Okay.
Yeah, I'll piggyback off of that question, Eric, in that I personally see a difference in what motivates an engineering student versus a basic science student. And so I think that engineering students typically really get into the business of wanting to do something that would result in a technology or an application. And it's harder to sell them on basic research.
Yeah. Okay. All right. Great. Thanks, Farron. Cindy, you're up next.
Great. So our team is a little smaller, so it's just me and Wallace, and I'm in mechanical engineering. Wallace is in biochemistry, cell biology, and we have worked together before. So, and UCSF and Stanford are really close, so we have a lot of meetings and in-person visits. Of course, it slowed down during the pandemic, but both before and after the pandemic, basically we're back to normal. I think maybe the most important thing for our team is that we have a joint postdoc who's a cell biologist, and she is immersed in my lab. So she sits in my lab and works with my students who are mechanical engineers, probably like at least half of the week or even like 80% of the time. And she also spends time in Wallace's lab and just like doing, the more cell biology side of things. So I think that is the key, kind of tying the project together and really knowing both the engineering side and the cell biology side. And then in terms of challenges in training, I think once we get someone on board, I think they are excited about the project, and I think that our arrangement between UCSF and Stanford has worked really well. I think maybe the challenges that we have is like recruiting people to work on this project to start with, because Stanford is a little weird organism. It's definitely far from any of the mammalian systems that people are more used to. So I think that I would be happy to get ideas from other people how to recruit students and other postdocs and trainees to work on something that's a little unconventional.
Yeah, you need to improve the social media platform for your organism.
If there's a platform that is not run by someone crazy.
Right. All right, thank you. Chris, you're up next.
Yeah, thanks. So our project spanned both experimentation by Kate Adamala and John Glass, and then the theoretical side, which I just mentioned. And I'll say that, I think in any sort of interdisciplinary effort, there's kind of two possibilities. The first possibility is sort of the collage or gluing or stapling together version, where you say, I have these skills, you have that skill, you do this and I'll do this, and then we'll sort of put them together in a paper. And then there's sort of a second stage, which is more along the lines of, I'll talk to you about what I do long enough that you really understand it, and you talk to me about what you do long enough that I really understand it, such that we can actually ask each other hard questions about what the other person is doing. And I think you just saw the example of this on our team where John Glass asked me a tricky question after my presentation. And I think that's really important for experimentalists to be able to ask theorists, well, what assumptions are you making in this part of the model? How does that really work? I happen to know this biological fact that seems at odds with the way that you're thinking about the modeling. And for the theorist to be able to say, what are the challenges in this sort of experimentation? Where are there sources of noise? What isn't being controlled for? Where do we have the most ability to manipulate? And so that each half of the team is really asking the other half difficult questions. And I think that goes for both PIs and trainees, and was something that was very apparent in our project. And then I think there's also, coming out of that, you get to a place where you start to co-design things, where you really say, okay, here's what's easy in the experimentation, let's build theory to deal with that. And then here's what's easy in the theory, are there any experiments that can kind of test that? And so there's really sort of an emergent third thing that comes out of understanding each other's work well enough. And then I'll say all of that goes for trainees as well. I think the other thing that is important on projects like this for trainees is that they really feel empowered to reach out to each other across teams and to PIs across teams. I think it's a really good sign when trainees are emailing PIs on other parts of projects and saying, hey, I was thinking about this, it seems to fall in your expertise, do you have any ideas? Emailing postdocs of other PIs and saying, maybe we could work on this together because it seems like a tough issue. And we had tons of that on this team. People in Kate's lab would email me and propose ideas and share drafts and ask hard questions, and there was sort of this general exchange going on like that. And I think that's a really good sign that the trainees have become invested and feel some ownership over the project.
Yeah, it's also a great sign that the PIs have created that comfortable environment for the trainees to reach out.
Yeah, so I have a question for Chris. And that I've always found it a little tricky to do experimental and theoretical work because, and not picking out one side or another, but in the example that I'm using, the theorist wants to use their tool, and perhaps their tool isn't the best tool to solve the problem. And so how do you have that kind of discussion so that you have a fruitful outcome? Because I think in some ways that can lead to delays in a project, right? Because you can go down the wrong path and then realize you need to switch your focus.
I couldn't agree more with that. And I think it's important for experimentalists to push theorists on this. I think it's also important for theorists to ask themselves, is the tool I know the best actually useful here? And I think things do go astray because theorists don't ask themselves that question. And so I think in a really good collaboration, theorists should be saying things like, I thought about it and all these tools I know just won't work for what we want to do. I'm aware of this other literature and it'll take me a bit of time for me and a postdoc to learn that and get up to speed with it and think about new ways to develop it, but it's something I've been meaning to learn anyway. And so if you're willing to wait a little longer while we kind of spin up with these other tools or this other sort of intellectual avenue, then it'll probably be more productive to us long term. But I totally agree that kind of honesty about, and I think that goes all sorts of directions, right? You can also say, is the experimental techniques we have in hand actually the right ones for testing the questions that are coming out of the theory if we're excited about the questions coming out of the theory? And so I think that sort of honest self-questioning and cross-questioning is really essential. I totally agree, Corey.
All right, thanks, Chris. Next up, Jeff.
Right. So had a lot of similar experiences to what you've already heard with regard to the teamwork aspects, and those date back to the pre-COVID and pre-UROL era when we were doing the synthetic yeast genome with colleagues really all around the globe. And that was also, for us at least, the pre-Zoom era. We didn't use tools like that effectively. So in that case, we had an annual meeting, which was just absolutely amazing. And I also remember as a highlight of that program, when we had two PhD students do the last year and a half of their PhD. They're coming from a chemical engineering lab in China. And they came and they just totally learned genetics. They had managed to build entire chromosomes just with, protocols we had sent across the globe. But they really didn't know that much about genetics when they arrived. So that was amazing. And I guess the other major thing I'd say about the sort of challenges is that the ability of different trainees to function in an interdisciplinary way just varies tremendously. So, I have some cases where it's a person where I try to get them to do the computational portion, and they say, and maybe this is the most appropriate thing, I'm just going to ask my colleague in the computational lab to do that part and, be a spectator. And then there are others who they want to learn how to do it themselves and they want to really immerse themselves in it. And so I think there's no simple answer to what's the best way. I think there's also a lot to be said to be, as a trainee, to develop yourself as a true expert in your domain area and simply be aware of the other dimensions that can be brought in from the interdisciplinary perspective.
So Jeff, I have a question about just responsibility of faculty in facilitating that, right? Because ultimately we have to educate the students to do the work that we're interested in doing that's going to facilitate pushing the boundary of science and engineering. But from an undergrad perspective, we tend to stick to the same sets of courses, and we on occasion throw an elective in there. So how much do you think we have to sort of rebuild or reconfigure how we educate students to facilitate them doing the things that you want them to do that perhaps they're not comfortable doing?
Yeah, well, I think that's a really important question. And I think we've seen a lot of activities in the undergraduate space, like iGEM. We did a course called Build a Genome at Hopkins for many years, and it's now been replicated in a number of other places. We have them going here at NYU now, which is very focused on training undergrads to do interdisciplinary work in synthetic biology, because synthetic biology is fundamentally, it's one of the things that attracted me to it was, it's this almost like a clash between the engineering way of doing things and the biologists, intuition that life will never yield to engineering, right? I love that about the field. And so definitely that course put a very high premium on making sure that the students learned how to do the computational part as well as the lab bench manipulations. And I think those kind of courses are super, super useful for undergrads to prepare them for this kind of work.
Yeah, great. Thanks, Jeff. All right, Vincent.
All right. So I think most of the points have been very well exposed. I can add 2 points specific to the team I was part. So I would say Cheryl Kerfeld, Giovanna Ghirlanda, and Millicent Sullivan are probably more protein engineers in the group, and then Christine Keating was more a physical chemist, also a chemical engineer, but physical chemist. And then Barbara was a social scientist, ethics. And me, I come from physics, but I do synthetic biology, and I'm still in a physics department. So it was a very nice group with very different background. I would say if you put the COVID aside, things went very well. I think that the people, the PIs, the young trainees, they made all the efforts so that things went really very well all along the way. We had meetings once a month. We were able to stick to those meetings. We've been serious about this. And the trainees were presenting, that's it, either postdocs or graduate students. And once again, I would say that the young people, the PIs, we made the effort, and it's very positive experience. You have to make the effort to understand how the other discipline they think, what they are interested in, and why. And it was really good. The second point I wanted to add, and I take the occasion for an advertisement. I am an instructor at the synthetic biology course at Cold Spring Harbor. It's every year, it's 2 weeks, and we have 16 participants there. And they don't come just from synthetic biology. We take a physicist, we take modelers, we take, and they are all together for 2 weeks, and it's very interesting. It works very well. And so just to let you know that the course exists, and there's not only this one, there are many courses where eventually you can send your trainees so that they develop other skills. Here it's 2 weeks. For 2 weeks, they are going to discuss with many other people, different background, and that's it. And I think one possibility would be to develop that type of experience. 2 weeks is relatively short, but still it's substantial. And it's incredible experience for most of the participants. I mean, the feedback that we get are in general very positive. But my conclusion of this is that developing maybe summer school or offering a 1 year immersion at the end of your college degree, for example, you have done 4 years of physics, and giving the possibility of a physicist to go to a lab of biology or chemical engineer, and for 1 year to learn something else would be a way to develop and to improve the multidisciplinary research, and especially the education. That's it. Thank you.
All right, great. That's a good plug for your course. But along those lines, so I think everybody agrees that Zoom has been vitally important in keeping information flowing between groups. Vincent, do you think your group suffered from not being able to meet in person?
I think so, a little bit. At the University of Minnesota, which was probably the same a little bit everywhere, we were closed a few months, and then we could come back slowly. We had to share the space, make some rotation, and overall for my group, which is relatively small, I think things went relatively well at that level, I would say. I have not seen major issues, at least with the lab members that I know. That's it. Yeah.
Okay. All right, great, thanks. Okay, so that concludes the multidisciplinary and training portion of this. So we've covered 3 of the 4 points now. I believe Corey is going to lead us into the final section.
That is correct. I think we are 3 minutes from the break. So I don't know if we want to do the break first and then come back, or Carmela, what do you have any suggestions on that?
If people need a bio break, this might be a good time. Does anybody need to drink water, or?
I think we all need a break, but
yeah.
I think the question is, Corey, I think the question Corey's asking is, if we have to sacrifice something on the schedule, is it better to sacrifice before or after the break? Meaning, do we sacrifice some of the scientific insight, or do we sacrifice some of the kind of the concluding remarks?
Well, are you going into the societal issues addressed, Corey?
So I'm going into societal issues.
Yeah, so that is kind of different from what we talked already. So it's a nice, we can wrap that scientific part here, have a little break, and then you can do the societal issues.
Okay. So
yeah.
I think so. We're going to break here, and then we're going to come back in 10 minutes.
Yes.
So just to make it clean, let's say 2:30.
Yes.
And just to be clear, this is not exclusively societal. there's also scientific issues. So there's still scientific discussion that will
Correct.
happen after the 2-minute break.
Correct.
Okay, there we go. So I'll see everyone back here at 2:30.
Giovanna Garlanda, who's part of a project involved in membraneless organelles. And I guess I just want to make sure that the National Academy takes note. The comment related to maybe a disproportional effect of younger faculty during the pandemic that were trying to learn new skills, and how Giovanna was part of a group where she was stepping into something new, and she may have disproportionately suffered because of that, as opposed to an older faculty that has, things more up and running in their labs and is not learning something new. So I just wanted to bring that up before we begin the last section, and thank you for that comment, Giovanna.
Yeah, I think that's remarkably important. I think that's a broader conversation about the COVID scar, not just on faculty, but students as well. So I think that's really important. Yeah.
Yeah, and I'll second that I would say was a big impact for myself as well.
Yeah.
That's someone who was 2 years in when it started.
So we are in the last vignette. And I'm going to forego the slides, similar to Eric, and summarize what we're trying to accomplish in this vignette. And that is, we are going to attempt to address 3 prompts. The first prompt is the science issue that you've addressed and the importance of that on society. And then the impact of social issues that need to be discussed in the context of doing innovative work. And then we're going to wrap things up with the major takeaways and themes from the workshop. And what I'm going to do instead of sort of giving you some text to read, is perhaps ask the discussants, the PIs, to give us feedback in terms of what they perceived as the important takeaways from the workshop, and perhaps this can lead to broader and bigger discussions. And so I'll start with an example. So whenever I'm asked what we do, I get typically 2 responses. One is that, oh, that's amazingly cool. I'm excited that you can do that, and the impact on society seems promising. And then the other is that this is remarkably frightening. And so I think that we sometimes forget to discuss the narrative that we're doing the science and engineering in. And that's important because the way society perceives what we do is important. And so with that, I'm going to go ahead and open the floor for discussion. And I'll just go through the queue like we did before. I'm going to change the order a little bit. And so I'll start with Farron, and if he's around, and then we can go through the other five, 6 people that we have. Okay.
So just so I understand exactly what you're looking for right now, because it seemed a little bit repetitive from what we did earlier.
Yeah. So that the question is, I'll put it a different way. From a societal vantage point, what are you going to win if you win? And what are the things that we need to discuss with regard to how this impacts society, positive or negative, that need to be discussed?
Okay. So let's see. From a societal vantage point, I think there's an opportunity to introduce the concept of chirality in biological, like, molecules and systems, and the role that they play in, like, biochemical molecular processes. One thing I actually didn't get a chance to talk about, but an important aspect of our broader impacts has been engaging with artists to help work with us to actually illustrate concepts of chirality from biology and then relate them to well-known and relatable aspects that people are commonly familiar with, mirror images, or even using simple examples of pointing out how our hands are achiral. So I think that's one sort of societal impact, is really imparting concepts behind chirality. I think a lot of our efforts are also aimed at really pushing the limits of highly conserved enzymes and molecular machinery, polymerases, ribosomes, translation factors, to do do unnatural things and encode unnatural molecules. And I think that then lends itself to really the development of powerful technologies, both biomolecular technologies, but also perhaps new applications or products that might emerge, like new types of materials that can be made from engineered translation systems, specifically new kinds of drugs that could address longstanding challenges of biotherapeutics, and more broadly, coming up with new ways to encode synthetic or abiological chemistries, but leveraging the power of genetically encoded biological systems, I think has the potential for broad impact on society from a fundamental biological understanding, as well as being able to produce novel kinds of molecules. So those, to me, would be some of the big societal impacts.
So in your own experience, have you, like when you describe what you do, have you had instances where someone is perhaps put off or frightened by what you're trying to accomplish because it's so different? And how do you address that?
Yeah, that happens. I recall being referred to as doing Frankenstein type of work. Jeff Bucco's familiar. When I gave a talk at NYU a few months ago, and his introduction to me showed a skit on Jimmy Kimmel that actually mimicked our work. So I've actually been exposed to it in a pretty significant way. But I think that's actually an opportunity to put innovations in science in the forefront and use that to actually connect with people and broader society, and explain to them why we do what we do from both the biological perspective as well as the perspective of how it might impact them and their futures. So I actually think those can be opportunities to get attention and then engage in conversation, and perhaps even inspire them to participate in some way, whether it's them, their children, or friends.
Okay, so initially, any press is good press, and then work that to your advantage. Okay, I got it.
Well put.
Okay. All right, let's scoot over to Cindy. Same prompts.
Yeah, I think in terms of the societal impact, I think we really hope that we can identify wound repair mechanisms that that we can boil down to some physical laws that we can potentially start applying to other synthetic systems, definitely synthetic cells, and maybe even micro robots, soft robots, to make them perhaps more robust. So if they can have the ability to self-repair, then maybe we can send them into the wild and they can self-repair. They can be in the harsh environment of whatever environment that they have to be in, they can repair themselves. And also the other kind of angle is that some of the synthetic cells are potentially used for biochemical manufacturing. So the industrial scale up processes can be harsh. And if the cells have the ability to self-repair, then we don't have to worry as much about replenishing these cells. So that's definitely more far out, but that's something that we think it's definitely possible if we can identify some of the rules of wound repair in these single cell organisms.
And so the interesting thing about sort of how you lay that out is that immediately I'm thinking runaway nanotechnology, right? And so how do you respond to, say, some comment like that, or perhaps even if you've already experienced things of that nature, what are your responses?
Just to kind of run away, so like some of the stuff.
Runaway nanotechnology, right?
Runaway nanotechnology.
Yeah.
Yeah. So that's a definitely tricky one, and I think that people that I've talked to, they recognize that we're still pretty early in, just trying to understand the single cell, and we're still pretty far from actually applying it to anything at this point. But that's always sort of the worry that, well, okay, if you are really successful and one day these things can go in one and they basically never die, then like what do you do? And there's definitely interesting discussion, but I don't think there is any like resolution to that. I think one of the sort of tangential discussion was if we give the cell or like the robot the ability to self-repair, we have to somehow have a kill switch that somehow we can kind of turn it off. But of course, it will take a lot more research to figure out how to couple the function to allow them to self-repair, but at the same time, kill it off if we think that is getting dangerous.
Yeah, this is a theme that's coming up more and more in NSF solicitations where they are interested in biocontainment. And so the kill switch aspect of it is becoming increasingly important, not just from the vantage point of you having discussions with people who are not necessarily in the field, but I think funding agencies are also moving in that direction. So, all right, let's go to Chris.
Yeah, so I think on the societal impacts, I think with any new technology, you always have some risk of unintended consequences. And some of those are very hard to deal with because it's just you introduce a new thing into the world, into a very complex system, and you don't know how it'll interact with all the other things, right? So predicting which technologies displace other things, initially understanding like the long-term impact of CFCs on the ozone layer, when you're just trying to introduce a new type of cooling, all of these things are very hard to predict in advance. And so that's a generally tricky challenge. But then there's this other side of unintended consequences, which is when you don't understand all the design challenges well enough, that simply in the process of trying to build the thing, you might produce problematic things along the way. And so I think that's very much more of the space we live in with a lot of biology. It's like someone handed us a very complicated steam engine that then we were trying to design for our own purposes, and we don't understand the whole thing, and so we start tinkering with it, and sometimes it explodes and does all these different sorts of things. And so I think the societal issue that we addressed in part of our project is if we have theory good enough to understand the design constraints, then we're more in the realm of thermodynamics, where we're building steam engines up from scratch, and we sort of understand the trade-offs, and we understand the hard limits, and we understand where things become problematic. And so then I think that decreases the likelihood that we don't exactly understand what we're creating every time we do something, right? So I think just knowing the design principles helps us build things very specifically, and that decreases a certain sort of unintended consequences. And so I think, for our team, I would argue there's, a lot of utility in basic understanding, because it gives us precise ability to design exactly what we want. Now, that's a long road to get to exactly what we want, but it then at least helps mitigate a whole set of risks that people might be worried about.
Yeah. So the interesting thing about at least that particular dialogue, Chris, that I took away was you gave the example of an engine exploding, right? And so that's and can be a poison pill for the development of technologies beyond that first iteration, because you have had a disaster. And so do you believe that there's a way for us to have this open, honest dialogue about failures so that we don't necessarily stymie development and innovation?
I do, yeah, and I think it's about those conversations, and then I think it's also about recognizing the sort of unique challenges here, and that many other technologies we build all the way from the ground up, very incrementally in tiny steps. And for this sort of engineering, nature has given us, things that have billions of years of evolution, and so it gives us a very advanced machine to start working with. And so it's just a much harder problem than certain sorts of totally bottom-up technologies we've built in the past. And so I think honest conversations about why that's so hard is also really important, about what the unique challenges are there, what the unique risks are for for tinkering with machines that are already so advanced because they have their own evolutionary history.
So I have an interesting comment from Jeff. Rather than read his comment, I want to go ahead and have Jeff chime in. So,
oh yes, well, this is a famous old saw. It is difficult to make predictions, especially about the future, variously attributed to Niels Bohr, Yogi Berra, and Mark Twain, which I think is kind of it's a rallying cry for this topic.
so while you're up, maybe you can share some of your own experiences with addressing societal issues.
Sure. Well, so again, through the Synthetic Yeast project, we, of course, we did attract a fair amount of attention along with our colleagues at the Venter Institute from the general public. So I guess one of the things I learned was that it was really important to be able to communicate some of the complexities of what we're doing and why we're doing it, and to do so whenever possible using analogies that anybody can understand. And so one of the things we developed with the SC2 was to what we call genome scrambling, where we put LuxP sites throughout the genome, and then we could turn on the Cre recombinase and make, a million different genomes from one starting genome. And so I found that a really effective analogy was that we had a deck of 5,000 cards, and that was the 5,000 genes that we were now able to shuffle in various ways. And so I think when you're on the verge of publishing something complex, interesting, but likely to attract some attention, I think it's really important to kind of prepare yourself with some ways of describing what you're doing and also why it's important before the reporters come calling.
Yeah.
So that and I've been through that now multiple times. Just like Farron said, we do get the inevitable comparisons to Dr. Frankenstein. I think that's probably the most important lesson. Also have benefited a lot from working with bioethicists. So we had Deborah Matthews, who I mentioned before, had worked with us on the synthetic genome project, and she's also worked with us on this what we call the big RNA project. She was incredibly helpful. She was not always. She wasn't like a lot of people will hire a bioethicist to be their protector against these things, but she was right in there, like challenging us, to defend why, what's the societal benefit of the work you're doing, and who are the winners and losers in terms of who's going to benefit from the technology once it gets out there. So that was really great, and we actually wrote a paper together on sort of the various dimensions of the social and ethical aspects of the work we were doing as a community of scientists around the globe trying to work together on this big project. So that was actually very enjoyable, and we've now we have this new project, the Dark Matter project, which isn't funded by NSF, but again, we're working with a bioethicist who's really in there in the trenches with us as we decide which mammalian genes to explore in the context of engineered mice.
Okay, outstanding. So the summary, make sure that you get in front of the narrative. tell people what you want them to know. And then the other component of that was to enlist people outside of your immediate space that are professionals in terms of ethics, policy, et cetera, to help you also see your blind spots.
Absolutely. And then last but not least, we basically had an incredible opportunity that we discovered during the pandemic, which was, they were getting ready to shut down all the labs, and the whole COVID testing thing had kind of imploded at the CDC. And so we realized that because we use super duper high throughput PCR for all of our projects, that we were in a position to massively scale up COVID testing, which we did. And this was not our plan at all, but what ended up happening was that we built the largest COVID testing operation in New York City over a period of a few months by teaming up with a local robotics company. And we actually, that company, Pandemic Response Labs, ultimately did 11 million COVID tests. So that too was in part, supported by NSF, because NSF allowed us to keep paying our trainees, and they formed a sort of volunteer team that deployed this technology during the pandemic. So it was a great way also to be able to tell the story of how the work we were doing actually had an immediate social benefit.
All right, I'm gonna slide Vincent in here because he tends to go last, so I don't want to keep you to the last person this time.
Thank you, correct. So
Yeah.
Very good. So in the ProtoCell project, we take the biomolecules and we use them out of their context. What we are trying to do and still trying to do, it's build synthetic cells, some synthetic cells that do not exist. And it's at least at the beginning very fundamental. Eventually we had ideas of applications later, but it's very fundamental at the beginning, and for sure fundamental science is very difficult to explain to the public, the general public. Why is it interesting? You are not trying to treat a disease or to solve this problem, right? So this is this effort of towards the public and explaining why fundamental science without direct application is necessary. It's necessary to understand how the molecules of life work, and necessary to interface those molecules with non-living matter, soft non-living matter, solid state non-living matter, to develop a potential, I would say, a potential to create new materials and new applications. And so I would say for at least for this project, very fundamental at the beginning. It's a tough exercise. Myself, I have a family, and each time they're asking me, what are you doing? I'm trying to explain, and it's not easy. It's a good exercise, but not easy at all. And I think that is one of the major points, and trying to, for example, to tell them that we are just at the beginning of exploring the potential of living matter, how it can be engineered and reshaped to create new applications, new material that will have a lot of potential after, let's say.
Yeah. So do you think, this is going back to one of Jeff's comments, that analogy would be useful in trying to explain how basic research will benefit society moving forward?
Yeah, absolutely. It's an exercise that we should do, taking examples maybe from other fields. And I'm sure that there are very good examples of people working on purely fundamental and a few years later developing extraordinary applications that have changed.
Radiation is a great example of that, right?
Exactly.
Yeah.
Absolutely. Yeah.
Okay. Alan, I'm going to let you go last. So I'm going to go to Mary, because I imagine you get called on a lot because your first name begins with an A. So.
Yeah. Okay. So in terms of societal benefits, I mean, I think, again, as Vincent just said, our project is very fundamental to start with, but I think we're having new ways to control the force generation by cells and to understand it, I think has, long range impacts for being able to synthetically generate force either by cells or in robotics kind of situations that Cindy was describing to start with. And I also think part of the long-term societal impact of what we're doing is by widening our understanding of biology across the evolutionary tree, which I think is really quite a few narrow branches right now, and that we need to broaden that understanding. And then in terms of the science communication aspect, a lot of what I would say has already been conveyed, but I guess the thing that I would add is that our team really benefited from the inclusion of science historians and artists on our team. And the science historians were particularly useful for understanding how language can shape that public perception. And so they looked at ways that words like synthetic or artificial have been, how their meaning has evolved and their connotations have evolved over time, where it might start out positive and then, take on a negative connotation over time. So I think being cognizant of the way language we're using can evoke a certain sort of emotional response in the broader public that doesn't have that context is one way you can sort of be careful about communicating things that may not even be the context that we're intending.
Yeah, and I think, Mary, the thing that's helped me is like just having discussions, right?
Yeah.
And then practicing doing exactly that, because I think that we lack the practice of having those conversations where we're trying to mitigate the negative outcome of.
Yeah, exactly.
Yeah.
And having folks like really involved in the team from the very early stages who can bring that perspective, because they're not thinking about the things you're thinking about in science every day, really helps to make sure we're not losing track of that. Yeah.
Outstanding. So Alan, you want to close us out, at least for this small vignette, and then we'll spend the last couple of the minutes just highlighting the takeaways.
Sounds good. I'll be very brief since I know we're running out of time. So, my team worked with a social scientist that's on the team as on the proteus cell as well, and we're looking at essentially how the public perceives synthetic cells. So we have a technology that is very, very new, very few people have heard of. I mean, I think us in the field sometimes even debate about what synthetic cells are. So I think the public really has very little understanding. Just to highlight So our social scientists does this public interviews with basically normal citizens. Particularly, I think it was in Denver. They pick individuals that they interview because this is a biomedical application that that we're aiming for, the synthetic neuron. There's a great amount of enthusiasm that whatever we create ultimately could benefit human health, and so I think there's less skepticism. I think one of the findings is that if the target applications are aiming towards human health, there's generally a acceptance of that technology, even though there's obviously the safety issues that Corey, you were bringing up, but they were definitely very enthusiastic about that aspect. So I'll just leave it at that.
Okay, outstanding. All right, so Farron had to drop off the call. He said thank you for a very engaging discussion. And so with the last couple of minutes, I'm just going to let the PIs just do this as a call out. You do it yourself. I'm not going to call your name. And, 15 seconds, 30 seconds, tell me what the major takeaways from this workshop were for you. You're up, Alan, so you can go if you want. Yeah.
Yes, this is my name. Start with A. I'll start. Well, this workshop has been great. I really enjoy learning about all the different perspectives from the different project team. We had this one NSF gathering before, but this is several years ago. So it's definitely great to see all the progress that's been made in learning of all your rules. So thank you.
Understand, Chris, you can go ahead.
Yeah, I mean, I think my main takeaway is sort of emerging synergies, interesting lines of parallel thought that were very apparent in this workshop, and just how much progress has been made over the last 4 years. So I found the rules that people talked about very interesting. I think there's really exciting potential moving forward in this area, and that was very apparent. It was nice to see so many sort of convergent parallelisms. So thanks.
Okay, Mary.
That interdisciplinary research takes you in unexpected directions, but also can lead to really transformative progress, like Chris was saying. I mean, just hearing how much has happened since the last time I talked to folks was really inspiring.
Very cool. Cindy?
Yeah, I echo everyone's comments that it's really exciting to see so much progress. I wish there were more of these events and maybe also involving trainees. I think that would be tremendous.
Vincent?
The variety of projects, living cells are incredibly inspiring, and we can see so many different approaches, all multidisciplinary and somehow complementary, and exploring different aspects of the ways we could build synthetic cells.
Outstanding. Jeff, can you close up?
Sure. Although Vincent just stole my tagline, because I was going to make the same point. But yeah, I think this was terrific, and I'm really thrilled to have been a part of this and the former NSF programs that have really pushed in this direction and illuminated some new rules of life.
Outstanding. So I'll turn it over to Carmela. We're done. Thank you. It was very fun doing this.
Thank you, everybody. Have a good one. It's a great session.
It was fun.
I think we're relieved of duty. Okay.
Yeah.
Yes,
See you, folks.
everybody's dismissed. Thank you. Bye.